Our first order of business is to talk about microorganisms, and you might have noticed right off the bat that I didn't say what are microorganisms, I said what are microbes, and the reason for that is that microbes include both microorganisms and non-living microbes, and so in order to be more technically correct, we say microbes instead of microorganisms, and microbes include organisms from all over the biosphere. They include members of what was historically known as Kingdom Onera, the bacteria and archaea. They include members of Kingdom Fungi, the protozoans, protists, and algae of Kingdom Protista, and the non-living entities, viruses, and prions that we will discuss later on as well. So it turns out that microbes are found in every kingdom of life with the exception of Plantae. They're found in Kingdom Animalia, Protista, Fungi, Protozoa, and the two newer kingdoms that were made from Kingdom Onera, Kingdom Eubacteria and Kingdom Archaebacteria. Now, that doesn't always mean that these guys are always microscopic. Members of Kingdom Animalia are all multicellular, as we'll discuss later on in the unit, but some of those members of Kingdom Animalia are included in the microorganisms because they spend a significant portion of their life, though they are multicellular, as microscopic or too small to be seen with the naked eye. Same thing with protists and fungi. They're not always teeny tiny. In fact, some members of Kingdom Protista, the brown algae, are quite large, and fungi, like mushrooms, are obviously visible to the naked eye. But that is what we're going to be studying, and we're going to be focusing on the microscopic microbes, those that are too small to be seen with the naked eye, which is why across the top of your screen here you see a series of micrographs, photographs taken with microscopes, which can make things quite a bit larger than their actual size, and something that we're going to be discussing later on in this unit. So putting a pin in that for now, let's talk about measuring microbes. So microbes, like all things in science, are measured using the metric system. Metric units you might remember from previous classes include the meter as a measure of length, the gram as a measure of mass, and the liter as a measure of volume. We primarily discuss length units when discussing microbes. We'll talk about how long they are, and because they are microorganisms, we use very, very small increments of the meter, which you can see here up at the top of your screen are the nanometer, which is 1/1000th of a meter in length. So a typical red blood cell weighs in at about 10,000 nanometers in length. So fairly large from a microscopic scale, as you can see, though very, very small, generally speaking. But if you compare that red blood cell to the E. coli next to it, E. coli is about 3,000 nanometers, or about a third of the size of the red blood cell. And that's typical of prokaryotes. They tend to be much smaller than eukaryotic cells. But even smaller than that, if you take a look at the inset, is the virus, which is pictured next to just a small portion of the E. coli cell there. That bacteriophage, which is actually a bacterial pathogen, is about 225 nanometers in length, and HIV, the causative agent of AIDS, is about 150 nanometers. Now, they're using nanometers here because that allows them to look at all of these microbes and cells without changing units. But really, when we talk about E. coli, we typically describe them using micrometers or micrometers. And so that 3,000 nanometer E. coli is typically going to be described as a 3 micrometer E. coli cell. So that's how we measure microbes. It's not a new concept, but it's just something that we want to just refresh ourselves on, because you're going to be seeing micrometers and nanometers and micrograms and things like that throughout the course. The other thing that we want to hit is how to talk about these microbes. You notice I said E. coli, and E. coli is actually the scientific name of that microorganism. because it turns out that we don't name microorganisms with familiar names like we do people or horses or cows or chickens or dogs or things like that. Microbes are always named with their genus and species epithets, and those genus and species epithets, as you can see from the table on your screen, are often sourced in one of two ways. Either the source of the name is an honorarium or an honorific to acknowledge the contribution of some scientist to the study of that organism, or the source of the name is descriptive. So if we take just one of these as an example, Streptococcus pyogenes, the genus name Streptococcus is descriptive of what these cells look like under the microscope. So to the left there, you're seeing a scanning electron micrograph of round cells arranged in chains. And if you were to describe those using microbiological terms, you would say that round are cocci and chains are strepto. So when we say Streptococcus, we're referring to cells that are round in shape and arranged in chains. So Streptococcus means round cells arranged in chains. Pyogenes means to generate pus. So pyo is a word for pus. Genus is a word you'll see all over the place in medical jargon. It means to generate. So you can have pyrogenic, pyogenic, all sorts of things like that. Pyogenic means pus forming. So that is a nod to the fact that when you get a skin infection with Streptococcus pyogenes, it tends to produce pus, which of course is a bunch of dead white blood cells that have come into the area to try to fight the Streptococcus pyogenes. So no common names, but we do abbreviate scientific names much like we do in any other aspect of biology. So if we want to look at an example of that, take this statement at the bottom of your screen. Staphylococcus aureus and Escherichia coli can be part of the normal flora of the human body. S. aureus is found on the skin and E. coli in the large intestine. So notice there, after we've spelled out the entire genus name, we can then abbreviate it with the first initial and a period. Now, just a note for you to keep in mind, if you are typing these names, your word processing program is likely to capitalize the word that follows that period, and that would be incorrect. The correct way to write a genus and species name is to capitalize the genus, leave the species lowercase, and always italicize them when you're typing. If you are writing the names out, you're going to want to underline them. So that's how we measure microbes and how we name microbes. Let's talk for a second about what we're going to study in this class over the next 16 weeks. And what we will study is microbes and the different ecological processes that they are involved in. In Unit 1, we're going to look at microbes, what they are, generally speaking. We'll look at the anatomy of prokaryotic cells, and then we'll skip a few chapters of the Tortora textbook, if that's what you've purchased, and we will move to microbial diversity and ecology. Then in Unit 2, we'll move into looking at microbial physiology, or metabolism, where we look at the Krebs cycle, the electron transport chain, glycolysis, photosynthesis, and how all of those processes occur primarily in prokaryotes. We'll also look at prokaryotic genetics, how they replicate their DNA, express their genes, how they exchange genetic information, and then we'll wrap up that unit talking about the medical and industrial applications where microorganisms can be used to accomplish some particular goal, whether that goal be producing human insulin for mass market use, or producing vaccines, or bioremediating an oil spill, or something like that. That will be the subject matter of Unit 2. In Unit 3, we'll hit what you're probably here for, and that is host parasite interaction. So epidemiology, the study of disease, looking at microbial diseases, looking at the immune system and the body defenses, looking at antibiotics and pathogenicity of different kinds of microorganisms. And then in Unit 4, everything will culminate in an application of the knowledge that you've been gaining through lecture and lab in a series of 9 case studies where you will be presented with 9 people who have contracted some disease. You'll be given their signs and symptoms, their test results, and you will decide what's going on and how to treat those people. So hopefully that'll be a really neat way for you to see why you've been learning all of the things that you've been learning throughout the semester, and will allow you to put that information to good use. Now, we study microorganisms for lots of reasons, and really, I mean, the primary reason we study anything is just, I think, because as human beings, we have this innate desire to know, to seek out the unknown and make it known. And so we study microorganisms for that purpose, but we can apply the study of microorganisms to microbial processes, right? We mentioned biotechnology in the previous slide, the production of therapeutics like insulin or human growth hormone or vaccines or things like that that are helpful medically. The production of commercial products like enzyme cleaners for your laundry detergent, or xanthan gum as a thickening agent. Also ecological interactions, nutrient fixation. Microbes are solely responsible for fixing nitrogen and getting it into the biosphere. They're responsible for a tremendous amount of the carbon fixation that goes on on the planet, and they're also involved in lots of bioremediation. Because they inhabit really strange and unusual environments, they know how to eat strange and unusual things, and we're going to talk about that later. But they can eat things like crude oil, TNT, arsenic, things like that. So those are some things that we're going to study. And then finally, as I mentioned in the previous slide, we're going to study human interactions, both the interactions of our normal flora or normal microbiota and ourselves, and also the interactions of disease-causing organisms. And I think that's where lots of people's interest lies, and I think that that's where most people have the most experience with microorganisms, is how they interact with them. And there are two basic human microbe interactions. One is the interaction between human beings and most all of the microbes on the planet, and that is one that is a mutualistic, or at least a commensal relationship where neither partner is harmed. And one really beneficial or mutualistic relationship between humans and microbes is the relationship between the normal flora or normal microbiota of the human body and the human host. And it turns out that in and on you, you have about 40 trillion bacterial cells, which may freak you out a little bit, but it's not meant to. They're absolutely essential, not only in preventing disease, but in promoting your good health. Just a note that when we say in and on the body, we don't actually mean inside the body. Any microorganism that gains entry past the epithelial tissue is considered pathogenic. When we say in the body, we mean inside of the gastrointestinal tract or the respiratory tract or the genitourinary tract, where they are physically housed within the body. A lot of them are found on the skin and the gastrointestinal tract, and it's estimated that 30 to 50 trillion bacteria are passed in every bowel movement. And as you can see from the scanning electron micrograph on your slide, your bowel movements are chock full of a variety of different microorganisms. So those are typically very helpful to you in your gastrointestinal tract. As elsewhere, they prevent the growth of pathogens. They produce growth factors that you absorb from your colon, like vitamin K. They help you to absorb liquid from your colon and prevent diarrhea. And they actually produce antimicrobial compounds, which inhibit or kill pathogenic organisms, which you contract by eating or drinking contaminated food and water. So they're absolutely essential to your good health, and we're learning more and more every day just how essential all of these microbes are. Now, of course, there are also organisms that are not helpful, and we consider those pathogens, right? So they are in a relationship with us where one of us is harmed, the host, and the pathogen or the microbe benefits from that relationship. And though those only comprise about 0.1% of all known microorganisms, they're obviously an intense field of study because of the amount of suffering and possible death that those can cause in humans, and our study of microbes has really done a lot to change the dynamics of the human pathogen relationship. As you can see from the graphs here, in 1900, the top 3 out of the top 10 killers of people were infectious disease: influenza and pneumonia, tuberculosis, and gastroenteritis. If you fast forward 100 years, all but influenza and pneumonia have dropped off of that list completely, and influenza and pneumonia has dropped to the 6th killer, though because of the emerging disease AIDS, we do still have 2 of the top 10 killers in the world infectious disease. So that's sort of an intro to the course, a little bit of information about microorganisms and how we talk about them and why we talk about them and why we study them. And now we're going to switch gears to the second big topic of chapter 1, and that is something called the golden age of microbiology. And the golden age of microbiology is a period of time spanning the mid-1600s to the mid-1800s where dozens of really important fundamental discoveries were made within microbiology that both assisted in the understanding of microorganisms as well as biology and science in general. Now, obviously, we don't have enough time to go into all of those, so instead, we're going to focus on just a few of those advancements within that 200-year period of 1600 to 1800, and then hit 2 more, which are considered part of a more modern second golden age of microbiology at the end. Now, some of these topics we're going to talk about just today. Some of them we'll revisit later on, but all of them will kind of lay the groundwork for what we're going to be studying throughout the semester. So to start with, we have an Italian gentleman by the name of Francesco Redi, who began the biogenesis debate with his work on rotting meat. So right off the bat, you know things are going to get gross when you start studying microbiology. Sort of put a pin in that one for a minute. We're going to talk about him in a little bit, but for right now, we're going to fast forward a little over 100 years to Edward Jenner, an English physician who is considered the father of vaccinology because of his systematic approach to utilizing cowpox injections in order to prevent smallpox, which, by the way, was eradicated in the 1970s using the same technique that Jenner came up with in 1796. So we will talk about Jenner again later on in the semester, but for right now, just as part of that golden age of microbiology. Nearly 100 years later, we get to probably the best known name on this list, and that is a French chemist by the name of Louis Pasteur, who was a chemist and sort of all things biology and chemistry related, so sort of overlapped those fields, but was trained as a chemist, and through his training learned lots of things that led him to start looking into diseases, and he identified a protozoan as the etiologic agent of a silkworm disease, which may sound strange, but that was actually a fairly important economic commodity for France, right? Silkworms, of course, produce silk, which is then sold and traded all over the world, and in the 1800s was a far bigger part of the French economy than it is today. So Louis Pasteur, and then fast forward just about 15 years to actually one of his biggest rivals, German by the name of Robert Koch, who is known best for his postulates. Koch's postulates define the criteria necessary to connect microbes and disease. And then we go not even 10 years forward to a very well-known name and not a very well-known man, Hans Christian Gram, who discovered the Gram stain procedure, and unfortunately for him had no idea the tremendous effect that his discovery would have, because he made it accidentally. He didn't really understand what usefulness it might have. In fact, when he published, he said, "I have therefore published the method, although I am aware that as yet it is very defective and imperfect, but it is hoped that also in the hands of other investigators it will turn out to be useful." And he could not have been more right. And now we fast forward just nearly 100 years to 1975 to begin the second golden age of microbiology with the work of Kohler and Milstein, who produced monoclonal antibodies, which are going to end up being very important in our discussion of immunology and the applications of immunology, which include immunological assays for things which range from disease identification, like COVID testing, to pregnancy tests. And then we wrap up our second golden age of microbiology with the work of Barré, Sinosi, and Montagnier, two French scientists who discovered the etiologic agent of AIDS in 1983 as HIV, human immunodeficiency virus, which, although it did not herald the discovery of a cure for HIV, has led to enough treatment regimens for HIV that today, in 2020, HIV is considered a chronic illness, provided you have access to treatment. So that wraps up the golden age of microbiology, and now we move from 1983 all the way back to the BCs in order to talk about a fundamental question, and that is, what is the origin of life? Now, of course, we are not truly going to talk about the origins of life. That's more of a philosophical question. What we're going to do instead is talk about the scientific explanation for where living material comes from. And the scientific answer to that question lies with Aristotle, the very famous Greek philosopher and jack of all trades, who lived in Greece from 384 to 322 BC and came up with something called Aristotelian abiogenesis, where he described that a vital force which exists within organic substances, not life, but organic substances, can combine with the vital heat of non-living matter and spontaneously generate living material. So abiogenesis meaning that life is produced from non-living matter. So the generation from not living, abio, not living genesis. And in his writings, he said that it was readily observable that aphids arise from the dew that falls on plants, flies from putrid matter, mice from dirty hay, crocodiles from rotting logs at the bottom of bodies of water, and so on and so forth. And this was the prevailing scientific theory until the 17th century. And in the 17th century, there was this radical change in the way that people thought, and it was ushered in what we call the Enlightenment period, right? So the late 17th century, the turn of the 1600s to the 1700s, ushered in this time where empirical evidence, collectible, measurable evidence, was the primary focus for understanding what was going on in the world around you. So in 1646, a scientist by the name of Sir Thomas Browne is looking into this idea of Aristotelian abiogenesis, and he thinks to himself, you know, there just really isn't any evidence. I can't measure or see or touch or hear or feel anything that would lead me to believe that there is such a thing as this vital force within organic substances that can combine with vital heat and cause the spontaneous generation of living matter from non-living matter. And he publishes these thoughts in the Pseudodoxia Epidemica, right? The pseudo doxology, the pseudo truth, this epidemic's false truth. And then he subtitles that Enquiries into Very Many Received Tenets and Commonly Presumed Truths. So he's throwing out something there and he's saying, I don't think this is true. What do we think, scientific community? And you might think that everybody would be like, yeah, no doy, Tom, like that's exactly right. That there's no such thing as this vital force and vital heat and blah, blah, blah. But in fact, the response he gets from the scientific community is really exactly the opposite of that. And one man, Alexander Ross, responds to him and says, to question spontaneous generation is to question reason, sense, and experience, right? All of these things that are fundamental to the Enlightenment ideas of your personal physical experience. And he goes on to say, if he doubts of this, let him go to Egypt, right? Let him go sense and experience this for himself. And when he gets to Egypt, he will find the field swarming with mice, begat of the mud of Nilus, to the great calamity of the inhabitants. So he's challenging Brown to go into Egypt, to use his senses, to use his reason, to use his experience in order to discover that Aristotelian abiogenesis is correct. And Alexander Ross gets a fairly good reception from the scientific community at large. However, there's one person who rises to this challenge of letting reason and sense and experience inform his opinion, and he's the one that begins our debate of whether or not spontaneous generation, Aristotelian abiogenesis, or biogenesis, life begets life, is correct. And that man is Francesco Redi, who you might remember from our golden age of microbiology, who worked with rotting meat, right? So he sets up an experiment because he believes that flies do not spontaneously generate from putrid matter, as Aristotle believed. He believes that flies lay microscopic eggs onto putrid matter. Those eggs hatch into maggots. Maggots, of course, metamorphosize into flies, and the cycle repeats itself. So he sets out this experiment where he has pieces of meat. He exposes some to the air, and not surprisingly, they grow maggots, and those maggots transform into flies. He covers some with cheesecloth. Because the cheesecloth can cover the meat, no maggots are there, and then when he takes the cover off, sure enough, maggots and flies emerge. And then he has this third control where he's covered the meat with paper. He leaves the meat covered and no flies emerge. And so he publishes these data, and his conclusion is biogenesis, right? There's no such thing as Aristotelian abiogenesis. These guys are not spontaneously generating as the meat decays. They are there because flies laid eggs, and flies come from flies, not putrid matter. Unfortunately for him, there's still a lot of spontaneous generation promoting science going on, and they say, yeah, it looks like you might be right. Flies don't spontaneously generate from rotting matter, but animalcules, which we're going to be talking about in a few minutes, animalcules, microscopic animals, can spontaneously generate. And people are so convinced of that in 1745, a guy by the name of John Needham, who is a proponent of spontaneous generation, sets up an experiment to prove the spontaneous generation or Aristotelian abiogenesis of animalcules. So he sets up this experiment by taking broth, boiling it to sterilize it, and then covering it. And when he does this, lo and behold, spontaneous generation of these animalcules occurs. So he publishes the data, and this is where we're going to see one of the most important aspects of publishing your data and viewed data, and that is that when you publish your data and you publish your experimental design, people are able to critique and make sure that your experimental design is sound and that the conclusions that you have come to actually are supported by the evidence that you've collected. And when Needham publishes these data, people look at his experimental design and they're like, you're a hot mess, man. You didn't boil this long enough to sterilize it. You left it open and then you covered it. And what you attribute to spontaneous generation could very well be attributed to animalcules in the air that fall into your broth. And very quickly on the heels of Needham's publication, Lazzaro Spallanzani produces an experiment to negate the results of Needham's experiment. So he takes a similar broth, he boils it, leaves it out, and lo and behold, animalcules exist. He takes another broth, which he has sealed, boils it to sterilize it, and because he keeps it sealed, no surprise to us, nothing grows inside of that flask. So he publishes these data and he says, see, there you go. If I don't keep the flask open to the air, it's sterile. If I open it, it's seeded by animal kills in the environment, and so there is no such thing as spontaneous generation. It's biogenesis all the way. And people say, negative, Spallanzani, you've kept out that vital force in those capped tubes, and so there's no way for the vital force to get in there and do the work that it needs to spontaneously generate these animalcules. Now, this may seem ridiculous to you, but what you need to understand is that at the same time these experiments were going on, people were discovering the need for oxygen that most all known organisms at that time had. And of course, you and I would be included in that list. If I put you in a bottle and seal it, I deprive you of oxygen, you're not going to survive. And so people said, no, spontaneous generation requires this vital force. You have kept the vital force out, and so that's why spontaneous generation did not occur. And it takes almost 100 years for somebody to come up with an experiment where they feel like they're ready to throw their hat in the ring and give it a go. And that somebody may sound familiar to those of you who have had physiology, Theodore Schwann, because of course Schwann cells were named for him. And in 1837, he becomes part of this biogenesis, spontaneous generation debate as a proponent of biogenesis. And so he sets up this experiment where he's got these sterile broths, and because he wants to allow the vital force in, he creates this air inlet from the broth through a tube and out into the environment. And just to make sure that he keeps everything out of his experiments, he places a flame under the air inlet, and that was the fatal flaw in his experimental design, because when he published his data, which included sterile broth which remained sterile, people said, yeah, but you added that heat, and though you allowed the vital force in, you damaged it by heating it, and so you still have not settled this debate for us. So 200 years, lots of different things. These are just 4 of the highlights that I'm pulling out for you, and still we have no clear answer. Is it biogenesis or spontaneous generation? And then in 1859, Louis Pasteur, who we mentioned in our golden age of microbiology, who is a proponent of biogenesis, sets up an experiment. And he takes some broth, he pours it into a flask, and then he heats the neck of the flask in order to create this curvature, which is called today a swan-necked flask. And then he boils these special flasks in order to sterilize them, and then he simply puts them out on the countertop, and lo and behold, those flasks remain sterile. When he takes those flasks and breaks the neck off of them, they grow contaminants, which would lead you to believe that the contaminants are coming from the environment. And he goes one step further and actually takes some of the flasks and tilts them sideways so that the broth can run into the neck of the flask, and then sets them back upright. And again, contamination occurs. So he shows that sterile broth will remain sterile even when air is allowed into the experiment, but as soon as exposure to the air is allowed, contamination occurs. And you can actually go, if you're lucky enough to go to Paris, you can go to the Pasteur Institute, and you can visit the Pasteur Institute and see 200 years later sterile broths that were prepared by Louis Pasteur in the 1850s and 60s to discuss this idea of biogenesis. And when he did this, this was the mic drop moment. This was the moment where biogenesis was supported, spontaneous generation was finally disproved, and today we don't discuss biogenesis, or excuse me, abiogenesis, Aristotelian abiogenesis, or spontaneous generation as an answer for where cells come from. We discuss biogenesis. Where do cells come from? Cells come from pre-existing cells. No matter how small they are, no matter how simple the life form, cells always come from pre-existing cells. Now we switch gears and talk about the tool of the microbiologist, the microscope, which of course is of utmost importance when you're talking about trying to learn about things that are invisible to the naked eye. You need a tool that will enable you to see those microorganisms in order to learn more about them. And one of the very first people to use microscopes, in fact, to make his own microscopes, was a gentleman by the name of Antony van Leeuwenhoek, who not only discovered microorganisms, but gave them one of my most favorite names ever, and that is animalcules, because when he looked under the microscope, he saw these things that were sort of a cross between an animal and a molecule because they were so small, and so he called them animalcules. And he was so excited about these animalcules that he wrote 520 letters to the Royal Society talking about these animalcules. He made more than 500 lenses, some of which resolved to smaller than 1 micrometer, 1/1,000,000th of a meter. He made 25 microscopes, which, when you look at his drawings, must have been able to magnify things up to about 500 times their size. 8 of those microscopes survive today and are capable of magnifying images up to 275 times their size. And he, as a lover of animalcules and an avid user of these microscopes, was resolutely opposed to Aristotelian abiogenesis. And as the father of microbiology, even Google thought he was cool enough to earn his own Google home screen animation on the 384th anniversary of his birth in 2016. Because some of van Leeuwenhoek's microscopes survive, we're actually able to see what he saw. And these two photographs that you see on the left-hand side of your screen are actually lenses created by van Leeuwenhoek in the 1600s that were used a couple of years ago to look at red blood cells and to look at the head of a louse. And the red blood cells, if you look at that picture and then listen to van Leeuwenhoek's description of his own blood, you'll see that what he was seeing is what you're seeing there in front of him. And he said, "I observed blood from my hand, which I found to consist of red globules floating about in a way like fluid." And if you look at that bottom picture and then compare that to these drawings that he himself drew and submitted in his first letter to the Royal Society, you can see that he was very capable of making out all of these intricate pieces of bees and lice and all sorts of teeny little insects, which of course are visible to the naked eye, but the detail that he was able to see was a result of his looking at them through the microscope. It's unbelievable to think that van Leeuwenhoek, the doting father of animalcules, was able to make all of the discoveries that he made using a single lens microscope, a replica of which is pictured on your screen. Now, this microscope housed the specimen at the end of a pin that was seated right next to one single lens, which was held close to the face and illuminated by candlelight. And not only is it amazing that he was able to see what he saw, but that when he saw it, he exclaimed with delight that no more pleasant sight had ever come before his eyes than these beloved animalcules that he so enjoyed looking at. He found rotifers and paramecia and algae, and he was so excited that he said the motion of most of these animalcules in the water was wonderful to see. He was just so excited about them. And then he did something really amazing. After he discovered these larger eukaryotic microorganisms, he actually discovered bacteria. Van Leeuwenhoek discovered bacteria using his microscopes in 1676. And he drew them, as you can see from this picture here. And these pictures on the right-hand side of your screen are actually pictures taken using surviving van Leeuwenhoek lenses. And he was so excited about these bacteria, he went on and on and on about them. he said there were so many of these very little animal kills, very prettily moving. And the biggest sword had a very strong and swift motion and shot through spittle like a pike does through the water. And then he described a second sort who spun round like a top and were far more in number. And he was just delighted, and maybe surprisingly, at all of these things which he actually found in his saliva, in scrapings of his mouth, and all over the place. Now compare these descriptions of what he's talking about here with this modern video taken under the microscope. See them spinning, right? Spinning and swimming around. Now, obviously, he would not have seen them at this magnification. This is a picture at 1000x, but he definitely was capable of seeing these microorganisms that today we call bacteria. So not only is he the father of microbiology, he's the father of bacteriology as well. He also discovered a scourge of the modern age, Giardia, and there's a picture there where you can see this Giardia under a lens that he produced, and then a picture of those Giardia using a stain and a little better microscope from today. Unfortunately, though he discovered Giardia, he found that it was so common in the stool samples and water that he was looking at that he had no idea that Giardia was actually a pathogen. And the discovery of Giardia as a pathogen actually doesn't occur until the late 1800s. The microscope, which was a huge, huge boon to empirical science, right, which was not happening in a vacuum. It was happening as part of the 17th century switch to empiricism and enlightenment, which was a major worldview and paradigm switch from the ancient philosophical writings as a way of understanding the world to data collection and publication of those data, very important, publication of the data, which could then be confirmed with separate study, which we saw very much in evidence in that 200-year spontaneous generation biogenesis debate. And the microscope provided scientists an objective methodology to study microorganisms, and it was foundational for the theory of biogenesis because it finally allowed people to understand what in the air was causing the growth of these animalcules. And it was foundational for something we're going to be studying later, the germ theory of disease, which is part of Koch's postulates, where an etiologic or causative agent of disease is connected to the disease that it causes. And none of that would be possible had van Leeuwenhoek not discovered, produced, and shared the microscope and his microscopic discoveries with the world. So we want to give a nod to that and then take a few moments to understand how microscopes work and kind of how they're used in a microbiology lab. So microscopes work by increasing the size of an image virtually. So very simple microscopes use a single lens, and you can see that on the left-hand side of your screen. If you just take a glass lens and hold it up, use a glass and hold it up to text, you can make that text appear larger. It doesn't actually increase the size of the text, which is why we say it virtually increases the size, but it does increase it to your perception. So on the right-hand side of the screen, you see how multiple lenses can be used to increase cumulatively the size of a specimen. So you see your eye looking through two lenses at a specimen. So the specimen is very tiny. One lens increases its size, and then a second lens increases its size further so that the virtual image is large enough that it can be seen with your eye. And that kind of microscope, which van Leeuwenhoek discovered, and which we see with our eyes, is called a light or an optical microscope. And light or optical referring to what is used to illuminate or allow that specimen to be visible. Now, today, the microscopes that we have are a far cry from the microscope that was created by Antony van Leeuwenhoek. And they look something like this. These microscopes, these light microscopes, contain more than one lens. As you can see on the right-hand side, several lenses which enable the magnification of the specimen to your eye, but they still use light, so they're still called light microscopes, but because they have multiple lenses, they're called compound light microscopes or compound optical microscopes. And the total magnification is the product of the magnification of the objective lens, which is housed over the stage or directly over the specimen, and the ocular lens, which is the lens that you perceive the specimen through. So on the right-hand side, you see a sort of a cutaway view of a typical student microscope and one that's going to be used in all of your lab activities. And then on the left, you see a picture of that. So you can see the ocular lenses at the top, and then you can see the objective sitting over the stage just above where the specimen would be sitting. And microscopes work because they increase the resolution. And it's kind of a weird term, right? But resolution basically means your ability to distinguish 2 points as separate. And if we look at this grid, this blue on white grid, and imagine what it means to be able to resolve this in better detail, it means to be able to look more closely at it. And when you look closely at this grid, what you find is that really each of those things that you perceived as a line is not a line, is actually a series of dots that are so close together that your naked eye could not resolve them as distinct. But at 10x magnification, making that line 10 times its size, you can now resolve each of those dots as separate from one another. So the naked eye does have the ability to resolve all of those lines as separate from one another, but it doesn't have the ability to resolve the distinction between each of the dots that make up the lines. And that may seem like a strange way of describing that, but what that means is when you take a visual exam, your ability to distinguish that top letter as an E means that you can distinguish each of those lines as separate. And as those things get smaller, they get beyond the resolving power of your eye, and you say that they're blurry. And they're blurry because you can't make out the distinction between each of the lines of that object. So when we're talking about magnification, we necessarily have to be talking about resolution, because you can't, well, you don't want to magnify past your resolving power, because when you magnify past your resolving power, then something gets blurry. For instance, this handsome little fellow here is my dog Mike, and I want to get a closer look at Mike, and I know you do too, right? Because you'd love to know what Mike looks like. So we get really close to Mike, but we can't see Mike because we've gone beyond the resolving power of the lens that I used to take a photograph of Mike. But if I can use a different lens which can both magnify and resolve the picture of Mike, then you get to see just how handsome Mike is, because you can both get closer to him and resolve all of the different parts of Mike as separate. So magnification and resolution are intimately related to one another, because without resolution, magnification just creates blurriness. So when we talk about microscopy, we talk about resolving power, which is the ability to resolve or distinguish 2 points as separate. And the shorter the wavelength, the greater the resolution. Now, unfortunately, the maximum resolution, or excuse me, the wavelength of visible light only goes down to about 390 nanometers, which is much shorter than the wavelength of an electron. So light microscopes have a maximum resolving power. The maximum resolving power of a light microscope is such that something that is smaller than 100 nanometers or 0.1 micrometers will not be able to even be seen with a light microscope. But still, that gives us a much larger range of visibility than we have with the unaided eye. So you can see at the bottom there, the unaided eye can only make something out that's about 100 micrometers in size, whereas the light microscope can see something that's about as 200 nanometers or 0.2 micrometers. Now, the smaller they are, the less detail you'll see, but still you'll be able to make them out. So on the left-hand side here, you see a light micrograph of some bacterial cells. So you can see them. We can see that they sort of have a rod shape, but we can't really make out much detail about them. If we want to have more detail, we need something that has better resolving power than light, and that something is the electron. So we move to a kind of microscope called a scanning electron microscope, and now we can see each of those cells in three-dimensional detail. We can even see the uneven surface of the cell. We can get further into that cell using something called a transmission electron microscope. And now, because we are looking at a thin slice of a cell, we can actually make out subcellular structures of that specimen. So there are lots of different kinds of microscopes. In this class, we're going to focus on three: the light microscope, the scanning electron microscope, and the transmission electron microscope. Light microscope uses light to illuminate specimens. It has the lowest resolving power. Scanning electron microscope and transmission electron microscope use electrons to visualize the image and have a much greater resolving power than the light microscope. And we'll talk about each of those in detail in a few moments after we talk about something that makes the resolution of any specimen better, and that is staining. And staining increases resolution because when you stain a cell, you increase the contrast between the specimen and the background, right? Just like that, black E on a white background is much easier, you can imagine, to see than a very light gray E on a white background. So staining increases the resolution by increasing the contrast and the visibility of a specimen, and staining of cells relies on the resting membrane potential of the cell, which those of you who have taken physiology might remember is always negative. So cells have a negative charge and opposite charges attract. So if we have a stain that has a positive charge, it will be attracted to the cell and it will stain the cell. Negatively charged stains are repelled by the cell and can still be used to provide that contrast, because while the cell remains colorless, the background that the cell is resting against becomes colored and the contrast is still there. So staining can be used in multiple different microscopic applications, and it's one that we use a lot in microbiology because it makes those very small prokaryotes much easier to see with our compound optical microscopes. So let's look at three kinds of microscopes. And as I mentioned, there are lots of different kinds of microscopes. We are going to look at three here, and that is the bright field optical microscope, the transmission electron microscope, and the scanning electron microscope. I have put at the bottom of each of these a picture of a paramecium so that you can get an idea of what this relatively large eukaryotic protozoan looks like when you look at it using each of these kinds of microscopes. So this doesn't mean that this is the best thing that this microscope could be used for. It just is giving you an idea of the differences in what you're able to see. So the bright field optical microscope is a compound optical microscope. So you have light shining up through a lens which condenses the light into the specimen, and then that light flows through an objective lens and an ocular lens where it magnifies twice, and you see a specimen that is flat, is not three-dimensional, and though you can make out the general shape and you can see little hair-like things on the outside of it, it's not super detailed. Now, a similar kind of microscope in the way that it works is the transmission electron microscope, though here it's transmitting electrons, an electron beam, instead of light. So if you take that bright field microscope and just flip it upside down, you start with the release of the electron beam from the top. It gets condensed. It flows through the specimen into lenses which magnify and collect the image, and then you view the image. So you need the electrons to be able to move through the specimen, which means that you have to have an incredibly thin specimen. And they're actually when you prepare a specimen for transmission electron microscopy, they're usually dehydrated and then they're embedded with a resin, and so that they maintain their shape, because they're then going to be sliced in first into semi-thin slices 1 micrometer in thickness, and then with this very special diamond knife, they're cut into sections of 50 to 70 nanometers thickness and then placed on a grid of metal so that electrons can transmit through them. Now, you don't need to know any of the details of that. I just thought you might be interested to know just how thin these specimens are. But because the electrons are passing through the specimen, you still see a two-dimensional image, but you see something that is much, much, much more detailed than you did with the transmission of a light through the specimen. Now, a scanning electron micrograph works very differently from a transmission electron micrograph, and as its name suggests, it doesn't transmit electrons through, it scans the surface of a specimen with electrons. And so in order to prepare a specimen for scanning electron microscopy, you have to first coat the specimen with something that will very easily give off electrons, and usually that's gold. So it's spattered with a very fine gold powder placed into the scanning electron micrograph or microscope. Electrons shine down onto the specimen, and that excites and sends off the electrons from the gold. So those secondary electrons are collected, and that allows you to get a three-dimensional image of that specimen. So it's not necessary that you slice through them, because you're not going to transmit the electrons through. You're just trying to get a look at the outside structure of that specimen. So you can see here the scanning electron micrograph enables you to have a three-dimensional view of that specimen. So they're useful for different applications, and depending on what you want to see and whether or not you have one of those microscopes available, you can use a light microscope, a transmission electron microscope, or a scanning electron microscope to get a variety of different details about a given specimen. If we compare these three microscopes, the transmission electron microscope is the one that has the greatest resolving power. So we have the light microscope, which has the least amount of resolving power, but still can resolve things to about 200 nanometers or 0.2 micrometers, so pretty good. Then we get the scanning electron microscope, which has greater resolving power and allows you to see images in 3 dimensions, and then the transmission electron microscope, which allows you to see much, much higher magnification and a detailed look through the specimen. So bright field microscopy usually is thought to be able to increase the resolution such that you can magnify something about 2,000 its size rather, and still be able to make out most things. Scanning electron microscopes are capable of magnifying a specimen 1,000 to 10,000 times their size, and transmission electron microscopes, because they have the greatest resolution, are capable of magnifying specimens 10,000 to 100,000 times their size. As I mentioned earlier, staining can be used to increase the resolution of a light microscope in order to see the cells against a contrasted background. And not only does staining increase the resolution, it also, if we employ stains together, allows us to learn things about microorganisms. And when we use microscopes to learn things about microorganisms, we call that microscopy differential staining, because it allows us to differentiate cells into 2 groups. And the most well-known of those 2 groups is thanks to the work of Hans Christian Gram, whose Gram stain procedure allows us to differentiate microorganisms into gram-positive cells and gram-negative cells. So we'll talk more about the Gram staining procedure in lab, but basically in any of these differential staining techniques, there's 2 stains involved, and there's a destain process. So one stain stains everybody. The destaining step destains a portion of the cells, and then a counter stain stains the remaining cells, so that at the end of the process, you end up with 2 different colored cells, like the round purple cells and rod-shaped pink cells that you see on your screen there. Now that is a picture of a gram stain. So I would say, if I was describing that, we have gram-positive streptococci and gram-negative single bacilli, and that in a clinical setting would be really useful because it turns out that antibiotics are prescribed based on the gram reaction of the cells, because a lot of antibiotics interact with cells based on the same properties that cause the gram stain reaction to occur the way that it does. So antibiotic use can be tailored to a particular infection based on the gram reaction of the infectious agent. It also allows you to diagnose infection and to characterize unknown microorganisms. Another really important medical test is the acid-fast stain, which is a differential test that we'll be talking about later on, which allows you to identify cells which have mycolic acid in their cell wall, and that can be a diagnostic tool for tuberculosis. Spore staining is a differential stain that allows you to identify spore-forming bacteria, which is not really all that important clinically, but it is incredibly important for food preservation and safety, because a lot of would-be food-associated pathogens are spore formers, and so we need to make sure that we kill spores in order to ensure the safety of the food that we're consuming. So those are differential because they differentiate between those types of cells. And then another related kind of staining technique, which employs more than one stain, is a capsule stain, which is really important because encapsulated bacteria tend to be much more pathogenic than their non-encapsulated counterparts. And we're going to be looking at something like that later on, where a encapsulated strain of Streptococcus pneumoniae is capable of infecting and killing mice, whereas the non-encapsulated strain is not. So that wraps up microscopy. A lot of that microscopy stuff is important on its own, but also is very important as you learn and try to understand all of the staining techniques that we're going to be looking at in lab. So hopefully some of this will be useful as you prepare to do some of those lab techniques.